In this video, we're going to talk about size exclusion chromatography, how it works, its different advantages and disadvantages, applications, and experimental parameters. Also known as gel filtration, it is a method for separating molecules based on their molecular size as they pass through a filtration resin. The main parameters that should be taken into account are the size of the molecules in the assay, the resin, and the buffer solution that it needs to properly function. The resin consists of a porous matrix of chemically and physically stable spherical particles, or beads, with properties that minimize absorption of biomolecules. The main objective of this assay is to obtain the purified molecules and use them in experimental analysis.
The mechanism of this purification or separation method is really simple. We have two elements. The first one is a solution, and the second one is a plastic chromatography column. The solution is a polydisperse solution, which means it has a lot of proteins from different molecular weight or polymers from different chain sizes. On the other hand, we have the columns, also called matrices, that are basically resin-packed columns that have a lot of beads on them. The beads on the gel in general can be made out of different materials, such as agarose, dextrose, and polyacrylamide. Long story short, when you pour the liquid into the column, small particles will enter the bead and consequently slow down because the path gets narrower as the bead goes, whereas the big particles will go around the beads and get down faster. Ultimately, the solution is loaded in order of different molecular weight or sizes. It's important to mention that the molecular mass of the smallest molecule unable to pass through the beads is known as the exclusion limit of the gel, and we have to remember that the gel has a limit, meaning it can be saturated. Finally, as we can see in the plot there, the molecules can be separated into molecular weight as the column volumes with their absorbance.
Now let's talk about the advantages and disadvantages of size exclusion chromatography. The first and most important advantage is that this technique is based on a very simple physical separation mechanism that can very nicely separate very large molecules from small ones. Also, we can introduce multiple solutions into the filter without them interfering with each other and without altering the biological activities of the molecules. Last but not least, its experimental parameters are simple, and they will be further explained in the next video. There are two main disadvantages to this technique. The first is that only a limited number of bands can be accommodated because the time scale of the chromatogram is very short. And lastly, the fact that there has to be at least a 10% difference in molecular mass to have a good resolution. What this means is that while it can separate big molecules from small ones, when molecules don't vary that much in size, then the technique is not that successful in separating them.
So, what are the main applications of the size-exclusive chromatography? The main applications of this technique is to separate different polymerized molecules by its molecular weight. And for example, it is used to separate hemoglobin from sodium chloride in a Cefadex gel. That is one of the gels that includes the size-exclusive chromatography. And more related to our case and of protein purification, a protocol has been successfully developed for the purification of an oligomeric enzyme. This is an extramitochondrial acetyl coenzyme A hydrolase that is extracted from rat liver, and it is purified using this technique that is size-exclusive chromatography. With size-exclusive chromatography, we can separate a lot of different molecules. We can separate sugars, proteins, liquids, asphalts, and other kinds of molecules just by their molecular weight.